Hello everyone. Today we are going to deal with biomolecules. Most of the chemistry students are losing marks in this particular chapter because they hate biology. That's why we are here, that's why we are studying maths. But in case of NEET students, they ace this particular chapter because this lot more talk about biology. Let me tell you a secret. This chapter is wholly related to chemistry, not biology. Although the namings are somewhat related to biology, but here we are cutting to pieces what we are going to study this in chemistry. Let's get started with carbohydrates. Carbohydrates are defined as the polyhydroxyaldehydes or ketones are the substances that generally give these on hydrolysis. So basically, either this can be of polyhydroxyaldehydes or ketones, or upon hydrolysis this will give aldehydes and ketones. And also it contains at least one chiral carbon. What is chiral carbon? The carbon which is attached with 4 different groups. Hence, it becomes optically active. Right. So, the general formula of carbohydrates is CxH2OY. X and Y can be of numbers. It starts from 3, 4, 5, 6 like that. Okay. But if you asking me, ma'am, only carbohydrates lies with this formula, I would say no. But the more appropriate word is that mostly, because this formula holds true for acetic acid as well. Okay, let's get back to the subject. Carbohydrates are classified into based on 3 types. The first type is based on molecular size. Here, carbohydrates have 3 types, that is monosaccharides, disaccharides and polysaccharides. We are going to study in detail about this type in a while. Let me take you through the other 2 categories as well. Based on taste, it can be of sugars and non-sugars. And based on their reducing property, this can be of reducing and non-reducing sugars. What it reduces? And you are saying reducing property. What it reduces? Carbohydrates which can reduce tolerance reagent. What is tolerance reagent? Ammoniacal silver nitrate. And filling solution. Those are called reducing sugars. Okay, if a carbohydrate reduces those 2 reagents, then that is reducing sugar, and which can't reduce those reagents, then that would be non-reducing sugars. For example, glucose and fructose both are reducing sugars, but sucrose is non-reducing sugar. Reducing sugars contain either alpha hydroxyaldehyde or ketone, or they contain a cyclic hemiacetal or hemiketal structures, or maybe they are equilibrium with that structure, so that it reduces tolerance and Felling's reagent. Basically, it should have a aldehyde or alpha hydroxyaldehyde or free ketones to get reduced. Okay, on to the chemical reactions of glucose. Glucose is a monosaccharide. Where we are getting glucose? The mother of glucose, where we can prepare glucose, is from cane sugar or sucrose. The formula of sucrose is C12H22O11. Upon hydrolyzing this cane sugar or sucrose, we are getting two same molecular formula compound, that is glucose and fructose. Both the names are different, but the molecular formula is same, C6H12O6. And where we can get glucose? It occurs in ripened grapes, honey, and most of the sweet fruits. Glucose is known as a dextrose because it occurs in nature, as also there is an optically active dextrorotatory isomer. Let's see about the chemical properties of glucose. The first reaction is with acetyl chloride, CH3COCl. Glucose with acetyl chloride in the presence of zinc chloride, it forms glucose pentaacetate. How will that look like? Glucose, which will have a aldehyde structure. We are going to do the structural elucidation also. And then OCCH3 4 times, and then CH2OOCCH3. That is glucose pentaacetate. Along with that, HCl will be produced. This reaction demonstrates the presence of hydroxyl groups, because wherever the hydroxyl groups are there, this aceto group will get attacked. This group will get attacked the OH position to form a acetate structure. So in total, I am having 4 acetate group formed, which means 4 plus 1, 5. So which means that 5 hydroxy groups were there in glucose. Okay. Next reaction is that treatment with PCl5. Reacting with PCl5, glucose gives pentachloroglucose. The structure goes like this: CHO with CHCl 5 times with CH2Cl. Technically it should be 4 times. Yes. So I just segregated the primary and secondary alcohols. Okay, this is pentachloroglucose. Along with that, it is forming POCl3 and HCl. We have seen the reaction with PCl5 with hydroxy groups in alcohol chapter, right? It just replacing the OH group with Cl. The same thing happens here also. It confirms the presence of hydroxyl groups. Total, how many hydroxyl groups are there in glucose? 5. Okay. The next reaction, that is reduction reaction. When glucose is treated with sodium amalgam and water, it forms sorbitol. The structure is given over here, sorbitol. This reaction shows that glucose can be reduced at its aldehyde group. You can see in previous reactions I just mentioned the CHO group, aldehyde group. But here that aldehyde group got reduced to CH2OH, which confirms the presence of aldehyde group present in the glucose. Then oxidation. Upon oxidizing glucose with bromine water, these two are very important, guys. So note it down. With bromine water, glucose forms gluconic acid. And with nitric acid, it produces saccharic acid. I am telling you the formula as well. For gluconic acid, CH2OH with CHOH 4 times and then COOH. That is formed when glucose is treated with bromine water. Gluconic acid is formed. And saccharic acid. Saccharic acid formula is COOH with CHOH 4 times COOH. Most of the students get confused in this reaction. Please make the note of it. Saccharic acid, which is having 2 COOH, because it is with nitric acid, it is doing an additional oxidation. But in case of bromine water, primary hydroxy group still remains the same, and aldehyde got oxidized to COOH. Okay, and formation of these acid proves that the presence of both aldehyde and primary alcohol groups are there in the glucose. Then formation of glucosone. We have seen this ozone formation in aldehydes chapter. What we have seen, when the presence of carbonyl groups, this phenylhydrazine structure is C6H5NHNH2. We have seen hydrazine simply also, right? And 2,4 DNP. So those comes under same category. What happens over here? So with aldehyde group is just react like this. CH double bond N NH C6H5. Water molecules will get eliminated, and here also N NH C6H5 and then CHOH 3 times, along with that CH2OH. This is glucosone, which has a characteristic yellow needle shaped crystals formation. This is a crucial identification test for glucose. Okay. Other than this, we have some structural insights as well. The straight chain structure of glucose explains most of the properties. The important test for straight chain is that HI with rest phosphorus. Glucose gives a 6-carbon system. That is hexane, N-hexane formation, which gives glucose is having a 6-carbon system. But these have failed to explain these 3 important observations. The first observation is that no reaction with 2,4 DMP and sodium bisulfate. Although that holds true for those straight chain, but these tests are fails. And then no reaction of its pentaacetate with hydroxylamine NH2OH, and existence of alpha and beta forms. Okay, so these 3 are important part. So what is alpha and beta forms of glucose? Before that, through these observations, what we have found is that glucose is not having a straight chain thing, it is having a cyclic structure. And also the equilibrium exists with the open chain form. Right, as you can see, D plus glucose is there, alpha and beta forms are in the equilibrium. You can clearly see the difference of alpha and beta form with OH groups. Okay, let's move on. And glucose is a 6-membered ring containing an oxygen atom in the pyranose form. Okay, I'll show you the glucose structure as well in a while. These properties are very similar to fructose as well. Okay, but fructose is having a 5-membered structure because the structure is differ in this point. Fructose contains keto group, but glucose is having aldehyde group in it. Okay, these are the key difference between fructose and glucose. You can see the fructose structure as well. Fructose is furanose structure, but glucose is pyranose structure, like this. Please work on this structure. This is very, very important. At what point the OH groups are differently attached, that is important. Okay, then there is a term called mutarotation. What is the definition of that? What does it mean? When these two anomeric forms like alpha and beta are separately dissolved in water, they undergo a specific rotation change till it becomes constant after some time. What is specific rotation? We know that optically active compound, when we are passing it through the plain polarized light, there will be a rotation caused by it. It can be of clockwise or it can be of anti-clockwise. Those values are noted. Those are the specific rotation of that particular compound. Here you can take, from taking alpha D glucose, are converted into the beta D glucose in aqueous solution and reaching a equilibrium point, but the specific rotation changes from 112 degree to plus 19 degrees. Okay, so this kind of rotation is kind of changes is called mutarotation. And another important term in carbohydrates, that is anomers. The question can be asked from this part, what is anomer? Which of the following is anomer? That kind of question you can expect. Anomer is very simple. It refers to a pair of stereoisomers which differ in configuration only around C1. That is more important. Only around C1 position, that is called as anomeric carbon or glycosidic carbon. This explains the existence of alpha and beta forms of glucose. Okay, as you can see, I am showing the glucose structure once again. The OH group, H and OH, differs in C1 position. That is changing in case of alpha and beta glucose. Okay, these are glucose, fructose are monosaccharides. Right? It can't be hydrolyzed further. It is a hydrolyzed form. It is, we are getting it from sucrose, that is a disaccharide. The next concept we are going to see, that is disaccharides. Disaccharides are nothing but when we are hydrolyzing it, it should give two sugars, two molecules of carbohydrates. They are disaccharides. For example, sucrose. It gives glucose and fructose. It is a non-reducing sugar. Note it down. Each and every point, note it down. It is a non-reducing sugar. Maltose, which is having the same molecular formula as sucrose, but it is a reducing sugar. and lactose. Formula is same, but it is also a reducing sugar. Sucrose is non-reducing, note it down. Okay, what about polysaccharides? Polysaccharides are the one which can give three or more than 3 molecules upon hydrolysis. For example, starch. Starch is a most significant polysaccharide compound. It is an energy source for humans, that we know. And what are the key characteristics of starch is that it consists of 2 main components, amylose and amylopectin. Amylose and amylopectin. Amylose is a straight chain that is having alpha 1,4 glycosidic bonds. Alpha 1,4 glycosidic bonds between glucose. But in case of amylopectin, it is a more complex structure, but point to be noted here is alpha 1,4 glycosidic bonds in the main chain and 1,6 glycosidic bond at the branching chain. Okay, so these points just note it down, which are very, very important. I'm just erasing it so that you can view that clearly. But these are very important. The questions can be asked from this. That's why I'm telling you these are important. When we eat starchy foods like rice, chapati, any form of rotis, bread, bun, or you eat pav bhaji, everything. Any kind of starchy food we eat, our body begins to break down it immediately. How it does, how the process works is that first starch is becoming dextrine and then it goes as maltose. See, the formula changes, dextrine to maltose, and then it got separated to glucose. The sequence. So how we can find the presence of starch? That is also an important question. The property of the starch is its reaction with iodine. With iodine, it will give characteristic blue color. Okay, that tincture solution, tincture of iodine, is nothing but you can check that color it will produce. It serves as this test is a key identification test for presence of starch. Starch does not reduce Felling's and Tollens reagent, but its reducing groups are involved in glycosidic bonds. That's why this is not reducing the reagents. Okay. The second important polysaccharide is cellulose. It is different from starch, where we saw that word cellulose. It's from plants, isn't it? So it is a structural framework of plant cell walls and is remarkably abundant. It is having 90 to 95% of cotton and 45 to 50% of wood. The structure involves beta-D-glucose unit connected by beta 1,4 glycosidic linkages. You see, 1,4, one from one position to fourth position. and then first position to fourth position. This is what they meant by 1,4 glycosidic linkage. Okay, so these are beta structures. You can see this is a beta glucose structure, and that's why we are saying beta 1,4 glycosidic link. Okay, then glycogen. Glycogen is often called as animal starch, which serves as a storage form of glucose in animals. We can find this in livers, muscles and brain as well. It is having a structure similar to amylopectin, but it is having amylopectin. I told you that is a complex structure, but still this is branched in case of glycogen. Next, we are going to talk about proteins and amino acids. Proteins are the complex molecules composed of amino acids linked through peptide bonds. What is that peptide bond? Let's say you are having carboxylic group and amine group, COOH and NH2. What will happen? One hydrogen from here. What will happen? OH from here and hydrogen from here gets removed, remaining CONH. That is what the peptide bond is. It is an amino acid. The breakup of protein is that peptide amino acids are linked through peptide bonds. Okay, and these amino acids share a common structure but differ in their side chains. That's what more important. Amino acids are classified into several categories. Let's see few important categories. Before that, these are colorless, non-volatile solids with high melting point and highly soluble in neutral solutions. And most important question can be asked is that except glycine, remember the name glycine, all other amino acids are optical isomers. All naturally occurring alpha amino acids belongs to L form. We talked about L form and D form in our isomers part, right? We talked about it clearly. So, except glycine, all other amino acids are optically active. This is the most important question you can expect from this amino acid part. Okay, so let's categorize these amino acids. The first category is amino acid with non-polar side chains. The simple ones like glycine. The structure of glycine is CH2, NH2 and another COOH group is attached to it. That's why I am saying this is optically inactive. And alanine, it's just a single variation, CH3, CH, NH2, COOH. This is alanine and this is glycine. Okay, so which are having non-polar side chains. And another example also valine, leucine, isoleucine. These are branched, but still they are not having any polar side chains. They are non-polar side chains. Special case in the first category is that proline. It is having a unique ring structure. It will be look like this. It doesn't fit in the general formula. That is also a additional point in case of proline. Okay. The second category is that polar but neutral side chains. In that case, hydroxyl containing side chains are serine and threonine. Sulfur containing side chains are cysteine and myothionine. Aromatic are tryptophan and triosine. This is important. And amide containing are asparagine and glutamine. Amino acids charged side chains. The charge can be of basic or acidic. Okay. The acidic side chains are aspartic acid and glutamic acid. And the basic side chains are lysine, arginine and histidine. But the name indicates, you can remember it, they are acidic side chains, and these are lysine, arginine and histidine are basic side chains. Okay, so while talking about the amino acids, the most important question is about zwitterions. Before that, you know about the amphoteric nature, which can act as a both acid and bases. This leads to the formation of zwitterions, where the amino acids carries the both positive and negative charges. At a specific pH, that pH is called isoelectric point, the amino acid exists as neutral zwitterion, showing a minimum solubility. Next, our concept is types of proteins. We know that proteins are built up of peptide chains, and peptide chains are formed between amino acids. Amino acids are nothing but which are having COH group and NH2 group. Okay, clear. So now we'll talk about proteins. Proteins can be classified into two: fibrous proteins and globular proteins. Fibrous proteins are have parallel polypeptide chains held together by hydrogen and disulfide bond, which making them insoluble in water, like keratin, which is in our hair, and myosin. In case of globular proteins, they form 3D spherical structures, are typically soluble in water. Example, insulin albumins. Important example. Okay, and protein structures can have 4 different levels: primary, secondary, tertiary and quaternary. Among these 4, primary and secondary are super important. Don't worry guys, I'll link this doc with you. I'll put the link details in the description box. You can download it and make use of it. Let's see. Primary structure. It is the most basic level, and you can see the structure over here. This is a covalent structure of a protein called primary structure, and it shows how the atoms in protein molecules are joined to one another through a covalent bonds. That is important. Primary structures are linked through covalent bond. All proteins on hydrolysis give amino acids, that we know. Particularly, these structure will give alpha amino acids. In case of secondary structure, secondary structure has In case of secondary structure, this level show how protein chain begins to fold into regular patterns. The first part is that alpha helix. Alpha helix will look like this kind of spiral structure. Coil. Chain coils around itself like a spring. Okay, like a spiral structure. And in primary, we talked about covalent bonds, isn't it? Here, hydrogen bond is also taking place. Hydrogen bonds are between every 4th amino acid to stabilize this structure. This we can find it in keratin in hair, which makes it strong and flexible. Second structure, that is beta-pleated sheet, it will be like this, like a sheet it will form, and this is here also the protein chain folds back and forth upon itself, creating a flat sheet structure. These sheets are particularly common in silk proteins, which gives us a unique strength. And these are having zigzag manner, and polypeptide chains are arranged side to side manner, as you can see, sidewise arrangement. The neighboring peptide chains are bonded together by a intermolecular hydrogen bonds. Intermolecular hydrogen bond. Next, we are going to see tertiary structure. Here, the tertiary structure is more related to of 3D shape. The final 3D shape of a single protein chain, that is about the tertiary structure. The various forces are come into play. For example, hydrophobic interactions push some paths inward while the polar regions stay on the surface. This level gives the protein like a hemoglobin their specific functional state. You must have seen hemoglobin, right? It will be like this. So, tertiary structure gives the hemoglobin and myoglobin have this type of structures. Finally, quaternary structure of proteins. The complex proteins are formed from 2 or more alpha helix structures. Alpha helix structure, we already saw that it's just a spiral thing, right? In case of quaternary structure, this structure indicates the way in which these alpha helix structures of a complex protein are associated with each other. That's what the quaternary structure implies. Hemoglobin, once again, it has a 4 subunits, which is a classic example for this kind of quaternary structures. Tertiary and quaternary of the complex structures, they won't be asking much question except the interaction part, but primary and secondary structures, especially the secondary structure interaction part, what is the structure and what is an example for it, are most important. Okay, then we are moving on to enzymes and their properties. What are enzymes? Enzymes are something related to catalyst kind of thing. Okay, so enzymes are the natural catalyst where we can see the enzymes. Enzymes are there in our body also. What it does is, when an enzyme meets a substrate, it forms a ES complex, enzyme substrate complex. This complex turned to be a intermediate 1 as a second step. Intermediate 1 then forms the product. Finally, the product breaks down and free enzyme and the product will be formed. Like very similar to our catalyst. What does it do? It facilitates the reaction. Right. Similarly, enzymes is also facilitates the reaction for the formation of product. Many enzymes are, most of the enzymes we can say with the name with ends with the A sound. Okay, maltase. We can see many enzymes name. Here also we have seen peptase, which used to break down our protein molecules. Right? Enzymes are needed in very small amount, and they are highly specific. That is, they catalyze a particular reaction only. Okay, they can't catalyze it everything. It's like a particular species. It's more selective one, and also it is very efficient. It can catalyze many millions of reactions per second. Specificity, it just follows the lock and key method. While we are saying about a lock and key, a particular key goes for particular lock, not for everything, right? Similarly, the enzyme behaves. It's suitable for the specific reactions or specific substrates to form product. It is a temperature sensitive, most work at the body temperature, around 37 degrees Celsius, and it is also a pH dependent. Around 7 pH it works. Okay, that's the important. Next we are going to see a glance about vitamins. In case of vitamins, the most of the questions asked as what are the water soluble vitamins, what are fat soluble vitamins. These type of questions you should be taken care of. First of all, what are vitamins? Vitamins are the organic compound which can't be produced by the body. We are supplying it by our diet or medicines for the maintenance of our body. The multiple deficiency caused by the lack of more than one vitamins are common in human beings. This condition is known as vitaminosis, avitaminosis, and there are two types of vitamins. One is which are water soluble. Water soluble vitamins are B and C. BC. You can remember it like water is a busy business. So BC, B and C vitamins are water soluble. Other than this, A, D, E and K are fat soluble. Okay, one you remember, another thing will be easier. Okay, that's it about vitamins. You won't ask much questions. If they are asking also, they will cover in this part. Finally, we are going to see about nucleic acids and then hormones. First, nucleic acids are polynucleotides present in the nuclei of the living cells. We are made up of cells. Plants are made up of cells. Animals are made up of cells, isn't it? So these nucleic acids present in the nuclei, that is the center part of the cell, in the form of nucleoproteins. They are the fundamental molecules which is responsible for their heredity. Why we are getting our behavior from our parents? Knowingly or unknowingly, most of the behavior we are getting unknowingly from the parents. We are getting the features from our family, parents or grandparents. How we are getting it? It's because of these nucleic acids. Okay, so because of DNA, we are going to see more about DNA now. So nucleic acids are responsible for heredity and genetic information in all living organisms. First, pentose sugar. That is one of those essential components in nucleic acid. That is a 5-carbon sugar. The second thing, that is nitrogenase base. The nitrogenous base can be of two types. The one is pyrimidine base, the another one is purine base. We are going to see that. Okay, so first let's talk about purine bases. Purine bases are double ring structures. It has adenine and guanine. Adenine and guanine. Adenine contains 2 nitrogen rings and guanine contains 3 hydrogen bonds with cytosine. Okay. When this nitrogen bases and pentose sugar combines to form a nucleoside. Okay. So nucleoside, which is equal to pentose sugar plus nitrogenase base. Okay, that is nucleoside. Yes. Okay. What about nucleotide? In case of nucleotide, it is made up of nucleoside plus phosphoric acid or another pentose sugar. Phosphoric acid or pentose sugar plus a nitrogenous base and then again phosphoric acid. So that is about nucleotide. So talking about nitrogenous base, I told you there are two categories, pyrimidine base and purine base. Purine bases which has adenine and guanine. Similarly, pyrimidines are having cytosine, thymine and uracil. You have to memorize this. You can remember like pyrimidines cut. Cytosine, uracil and thymine. And this is AG. Aged cut. Can remember it like that. Okay. So in that case, the uracil, U, it's only present in RNA. Rest of the things are present in DNA, but in case of uracil, that is present in RNA only. Okay. Then, what is the difference between RNA and DNA? These are the general differences. Most important point is what is the pyrimidine base and what is the purine bases present in DNA and RNA. First, DNA means 2-deoxy-D-ribose. The name, of course, that is important. RNA is D-ribose. Pyrimidine bases present in DNA, cytosine and thymine. In case of RNA, uracil and cytosine. Purine bases present, adenine, guanine. In RNA also, adenine, guanine. Structure, in case of DNA, that is double-stranded helix. Like you have seen the structure like this. You can see the structure like this. For double-stranded helix in case of DNA. And RNA, that is so simple, single-stranded structure. Location is DNA present as the main nucleus and RNA present in nucleus plus cytoplasm. Replication, it is self-replicating, and RNA, that is not self-replicating. Size, DNA size is larger, but in case of RNA, that is smaller. Stability, more stable, but RNA is less stable. Out of this, the first 3-4 points are very, very important. Okay. Then, another type of question can be asked from here, that is from this rule. Chargaff's rule. It either can be NEET or mains, advanced, any other examinations, this is important. The first rule is amount of A, adenine, which is equal to thymine, and guanine, which is equal to cytosine. That is first rule. A equal to T, G equal to C in case of DNA. Second rule is purine basis, which is equal to pyrimidine basis. Okay. Next thing you have to remember how it is getting paired. A, that is having two hydrogen bonds in it, A and T. T is having two hydrogen bonds. G and C, guanine and cytosine, which is having three hydrogen bonds in it. In case of RNA, thymine is not there, so uracil comes. Okay, A is equal to U. So we'll see the questions in a while. You have to remember this adjig. Adjig, you have to remember this. Okay. Then, what are the biological functions it is doing? First, as I said, replications, DNA copies it is producing, and then protein synthesis, transcription. Transcription is DNA to RNA. Translation is something like RNA to protein. Translation, the mRNA directs the protein synthesis by this process. That is called translation. Transcription, that is the process of synthesis of RNA and DNA in the cytoplasm of the cell. Genetic code, the linear sequences, A means next to that, T should be there. That kind of sequence is also that biological functions significance. Then, mutations. What happens in mutations is that the change in nitrogenase base sequence of our DNA molecule which lead to the synthesis of the proteins with an alternate sequence of amino acids. You must have heard this term, mutations, in coronavirus period. Second variant, third variant. Because of this changes of nitrogenase base, these changes of the sequence basically, which is responsible for the protein structures change, the altered amino acid sequence has been formed. Okay, this causes the genetic disorders. Okay. Let's see some problems related to this nucleic acid because that is most important concept of this chapter. Calculate the number of hydrogen bonds in DNA segment containing 30% of adenine. Total number of nucleotides they have given, that is 100. Okay, so we know that adenine, they just given. As per the first rule, A is equal to T, right? So which means both are having 30%. Then, how much of the purine base is there, that much of the pyrimidine base is also there. 30% A and 30% T is sorted. But they are saying total number of nucleotide is 100. So remaining 40% is there. That belongs to G and C. So for finding hydrogen bonds, number of hydrogen bonds which is equal to A T pairs into 2, because that is having two hydrogen bonds in it, plus GC pairs, that is having how many hydrogen bonds? 3, isn't it? So 3. So total it becomes AT pairs is how many? It is pairs I'm talking about. Okay, so 30 into 2 plus GC pairs 20. 20 into 3. which is equal to 120. So number of hydrogen bonds which is equal to 120. Okay, so that is the answer to this question. The next question is if a DNA segment has 20% cytosine, predict the percentage of other bases. 20% cytosine. So which means that C which is equal to G, that is 20%. First rule of Chargaff. Remaining 60% is there. That will be A which is equal to T, 30. So 30 A and 30 T, so total 60. Okay. That is what the answer to this question. The total it became 100%. Okay. So separately also they will ask you the questions. The last part of this chapter, that is hormones. That is a simple topic. The hormones are the chemical messengers that can be classified into three main types. First one is steroid hormones like testosterone, estrogen, cortisol. These are steroid hormones. These are fat-soluble molecules that easily cross cell membranes. It is responsible for control development and metabolism. The second one is peptide hormones. Examples are insulin and oxytocin. Water-soluble proteins that walk through the cell surface receptors. It controls various metabolical processes. The third type is amine hormones. Examples are tyrosine and epinephrine. It is derived from amino acids, as the name indicates. It controls the metabolism and stress responses. Okay, just the classification and which is soluble in water. That's more important. Okay, let's solve some questions, some previous year questions, so that you will get an idea what kind of questions can be asked from this chapter. Okay, so the first question is that your polypeptide has 8 amino acids. Calculate the total number of polypeptide bonds present. So in a polypeptide means what? Peptide chains are attached or linked together, right? So number of peptide bonds is always 1 less than number of amino acids. Remember that. Because when they are linking it, that becomes 1 less than number of amino acids. That is a trick to solve this type of question. Number of peptide bonds is always 1 less than amino acids. So they have given 8 amino acids. So how many peptide bonds will be there? 7. 7 peptide bonds. Very simple question. Then, which of the following statements about enzyme is incorrect? They are highly specific. Yes, they possess both active and allosteric sites. They are predominantly proteins. They can be used indefinitely. We know that enzymes are highly specific. Yes, enzymes have both active and regulatory sites, that is allosteric sites. They are predominantly proteins, but they can't be used indefinitely. They are more specific. They can be denatured by heat or extreme level of pH. They may be inhibited by various substances. They get degraded over the time also. So option D is the right answer to this question. Then, there is an advanced ECPC question. We have to just match it. Column A with column B. Column A, insulin. Which hormone that is? It is a peptide hormone. It is right. And thyrosin, it is an amine hormone. Let's say this is A, B and C. This is B. And estrogen, that is a steroid hormone. Then this type of question about zwitterion electron or zwitterion ion or isoelectric point. Very simple question. Listen to this. Calculate pH of isoelectric point of glycine if pKa is equal to 2.34 and pK2 which is equal to 9.60. So what is the answer to this question? So first, isoelectric point Pi, which is equal to pK1 plus pKa2 whole divided by 2. That is 2.34 plus 9.60 divided by 2. That is 11.4 by 2, 5.97. That's it. Therefore, this glycine will have a zero net charge at pH 5.97. Hope this session was helpful to you. Practice more and more problems. We are going to put practice papers also. Please stay tuned. Practice more.